Okay, so welcome to our final presentation. This is group 27, and our project was about bioplastic production from agricultural waste, and also involving engineered microbes. So our idea revolves around producing biodegradable plastics using engineered microbes that convert renewable resources, particularly agricultural waste, into environmentally friendly materials. This approach leverages synthetic biology to create and optimize microbial systems for efficient bioplastic production, addressing both waste management and plastic pollution issues. So, what are bioplastics? Bioplastics are plastics derived from renewable biomass sources such as vegetable fats, oils, cornstarch, or agricultural byproducts. And unlike conventional plastics made from petroleum, bioplastics are designed to break down more readily into the environment, reducing pollution and reliance on fossil fuels. So, in order to produce bioplastics, we need to involve engineered microbes. And the definition of engineered microbes simply is engineered microbes are microorganisms genetically modified to enhance or introduce new metabolic pathways in the context of bioplastic production. These microbes can be tailored to convert various forms of biomass, particularly agricultural waste, into biomass. So engineered microbes can be utilized to produce biodegradable plastics from renewable resources. And so the main maybe one of the main ideas that we used to come up with this project is that in Africa, the common economic activity is agriculture, and so the idea was how can we manage the waste from the agriculture. So there are four main generations of where we can get this waste. We have the first generation, second generation, third generation, and fourth generation. So in the first generation includes various food crops such as wheat, corn, rice, and sorghum. They are directly utilized as primary feedstock for energy generation and product manufacturing. Second generation comprises of lignocellulosic waste such as sugarcane, bagasse, wood chips, crop residues, and organic waste. The third generation entails of microalgal biomass, and on the fourth generation, we have biomass from metabolically engineered species such as bacteria and algae. Next slide. So we have some of the advantages of bioplastic production. Like first, it tackles key environmental and sustainability issues, reduces carbon emission into the environment, it reduces pollution with small carbon footprint. It is easily degradable, hereby having a positive feedback to the ecosystem. It is vital for reducing plastic pollution, minimizing fossil fuel use, and lowering emissions. It is clean, safe, and eco-friendly. It has an advantage over other technologies, but it is not without the disadvantages. Bioplastic production has some of the disadvantages, which entails high production cost, limited scalability and availability of feedstocks, competition with food crops for land and resources, potential environmental impacts, e.g. water pollution and land degradation. Number 5, we have lack of infrastructure for collection and processing of biomass. And lastly, limited range of bioplastic products. So I think the next presenter can continue from there.
Okay, thank you, Simon. With these advantages, the challenges from bioplastic, that's why we need to produce a plastic that give us the desired products we need. That's leading us to a polymer we are going to use. We have polymers, which is our starters for our plastic. The polymer we are using is PHA, and what we are trying to do is a copolymer, make is a copolymer, because PHA is more like it has also monomers of its own. So some of the other bioplastic, we have APL, we have BNC, we have BPA, and we have PLA, which are all biopolymers which are used for production of plastic. But PHA is used because of its wide versatility and easy degradability. Now, the solution we are trying to make is engineering our seed necator, which is our chassis, to produce the PHA, which expresses the PHAC gene, which will give us our desired property. Which desired property are we looking at? Good flexibility and uh, high, high tensile strength. Now, our Cinecator can actually make a copolymer of its own, but the copolymer is making is having a more of the 3HP monomer, which means we need more of 3HHX monomer. Now, how do we do this? We are trying to create a promoter. The promoter that creates this PHAX in Cinecator is not strong enough to trigger the gene to express the gene to our desired level. So we bring a stronger inducible promoter so that we are using an inducible promoter so that we can produce it, tune it to our own use, to trigger it to our own desired need. Now the this thing we are going to use is the promoter we are going to use is E. coli promoter, which is the P-TAC promoter, which is a stronger promoter on its own, and it is inducible, which we use to replace the promoter in the necator. Now, the SACB marker is used because of its special advantage, as it allows counter selection after the incubation of the construct, which means, and it also uses this thing as a marker. It also uses our biomass as a source of nutrients, which means we are still using agricultural waste for our marker. Now, the plasmid with the promoter is introduced to the Cinecator cells by conjugation, and the SAGB marker counter selection will kill the cells with SAGB marker, while those that has the integrated genome will survive as toxic waste released as byproducts from the SAGB. We kill those ones that are not integrated, that doesn't have the plasmid integrated into them. We make a franking sequence which helps us to target the particular gene, that's the PAC1 gene that we are trying to construct. We help us to target it directly so that the gene will not be going to another location we didn't send it, so that the plasmid will not go to another location we didn't send it. Then after that, we do our DNA sequencing to also confirm if the promoter has been replaced successfully. Also, we have to note that after doing all this, we have to confirm that if the promoter is not actually induced into our gene, it didn't go to where we send it, we have to restart the process again. We go again with the process so as to get our desired product. And also we have to look at the growth and the nutrients of the microbe which is being used, the Cinecator, to see how fast it grows, even as we induce our plasmid, the new promoter, so that we confirm that the promoter does not affect the growth of of our microorganism. Now, with this, I will give the mic to the next person.
Okay, thank you very much. So moving on, the sole purpose of bioplastic production from agricultural waste is to create a sustainable, renewable, and biodegradable alternative to traditional plastic, that is petroplastic. reducing dependence on fossil fuels, mitigating plastic waste, and promoting a circular economy. So moving on, without further ado, this brings us to the advantages and disadvantages of bioplastic production from agricultural waste. So number one advantage is high versatility as a result of induced promoter use. This means that engineered bioplastic can be tuned to desired level. Since induced promoters are used, we can make a bioplastic of choice. Then the rate of 3HHX can be controlled. So the second advantage is high degradability. This means that degradability is quick in PHA or like the traditional plastic, which takes hundreds of years to degrade. Number three advantage is engineered copolymer have a good tensile strength, mechanical and physical properties. The number four advantage is that the engineered PHA helps to also reduce agricultural waste as the waste products are used for feeding microorganisms. So disadvantages now. Number one disadvantage is the starting materials might be competing with agricultural land. This means that if there's no mass production of the starting material planted originally as crops in place of agricultural waste for the purpose of using it as waste directly without going through the process of agricultural waste. The second disadvantage is that engineering the promoter might be a tedious task with problem of flanking sequence. All right, so with this, I'll give it to the next team.
All right, thank you very much. We talked about can you hear me? So we talked about why this we are making a bioplastic, and we want to change from the normal plastic to bioplastic. But then 1% of the world's plastic production is just bioplastic, just 1%. So our dream of bioplastic looks far, but with our projects we can bring it nearer. So why is our plastic better? So we have different polymers used produced from bacteria used to make bioplastic in the process of bioplastic production. So why is our PHA better? We chose it because of its high versatility and it degrades easily. Our selling advantage of our bioplastic is what it degrades easily, and that's what we want. We want a sustainable and more efficient ecosystem in which we use our plastic and we don't litter it around to the environment, which will take years to degrade, which will litter the environment and cause a lot of adverse effects on the environment. So we have BNC, also another polymer from Acetobacter, a bacteria, but it requires very specific growth conditions. So if we want to scale up our production of bioplastic, we need to make it easy. We need the starting material to be easily available. This one, we are using starch. We need the glucose from the starch. An example of the agricultural food crop we are using is rice. So after rice, then also there's another polymer, PLA, that's polylactic acid, which is also good, but requires a polymer to be blended with. So when we mention engineering microbes, we also put into consideration the safety, the risk adhered to it. So we realize that there could be allergic reactions due to the modified pH structure. So we will test the allergenicity of the pH before we produce this out for the populace, and also we would make sure the production is in a contained environment, and also the toxic byproducts being gotten would be properly disposed, because that's one of the disadvantages of bioplastic. The toxic production that is gotten are mismanaged, but we assure that the toxic bioproducts will be properly disposed. We are a group of diverse members, but we came together we are joined together with the sole purpose of engineering a microbe to produce a better bioplastic with better durability and more use, and scale up the production of it. So, summary, our solution aims to not only benefit the economy, because these agricultural wastes are something in abundance in West Africa here. And also we need to reduce the carbon footprints. The world is on a race for reducing our greenhouse gases. So we also aspire to join the race by reducing our carbon footprints. When we reduce our petrochemical plastic, when we start to embrace the use of our plastic, we'll see the better efficiency and the better sustainability it brings to us. We need to also minimize pollution, because pollution is a very, very, very great risk to our planet. And we need to we aspire to build an ecosystem that is self-sufficient, sustainable, and scale up production. As I mentioned earlier on, we need to scale up this production of bioplastic with much desired effects. So thank you. These are reference. Thank you very much. We hope you enjoy our presentation.